It was fun. Oh, it was Mark. It was Mark. Yeah, met someone for breakfast. And he saw you was Simpsons. I was like, it's not like.
Testing, testing, testing.
Yeah. Sure.
3 times. It was so
Evan, did you watch playoffs last night?
I did.
The NFL this year has not disappointed.
It's fantastic.
Amazing.
Both teams are wanting to go and watch.
I'm hoping the Super Bowl is just as good as the playoffs have been. The playoffs have just been incredible.
Yeah, it kind of sucks that it's in LA, but whatever. I'd rather it be
SoFi Stadium is a beautiful stadium.
brand new too. So
That new stadium out in Las Vegas is probably just as nice.
I just want to be at some seats. I want Joe Burrow to win.
So does my daughter Stacy. She thinks Joe Burrow is the most beautiful thing to ever land here from heaven.
I know it. I mean, we're talking quarterback. I mean, Jimmy G got the best quarterback out there. So
Dude can ball. Bro, dude can ball. Dude can ball.
I mean, nothing like winning a national championship.
Yeah, no,
And then
when you figure where they were last year, I mean, the Bengals stunk last year. Absolutely stunk. And now look at them. They're in the Super Bowl. Amazing.
Nah, he did. He does that.
Stafford kicking around with the Lions all those years. People knew he was good. People knew he was good. He finally got a chance to show that he was good, and he's shown it.
He just needed Aaron Donald, Odell Beckham, and Akers, and the whole team basically.
He needed targets. You need good players around you.
Exactly, just not the line.
Yep.
Anybody but the line.
Yep. Okay, let's go ahead and get started. Don't forget, tomorrow we have our first lab for the section. Those of you in section 1, we'll be concentrating on renewing, rejuvenating your ability to recognize structures underneath the microscope. So this will be a chance for you to look at slides, draw some pictures, get reacquainted with structures. I talked today to Beth Ulrich about using the EKG machine and the metabolic cart because it's her office essentially, and she was, yeah, go ahead. The only issue is that the EKG system is completely different than the one I knew quite well. So I've got to relearn. Did any of you know, did Mitch make a video of how to use the new EKG system? I know there's a video on the metabolic cart. Crumbs. Now, Brackett, your name came up as someone who knows how to use that system.
That was a lie.
That was a lie. Okay.
I took the EKG class with a couple other HCS students, and we learned how to put the stickers on each other, but never how to press the button.
Great.
Someone's got to know how to do it.
If anyone knows how to use it, I will pay them. to come in and help us out with that lab. I will pay them. Seriously, I will pay them. That system I was looking at and saying, iWorks, I was able to figure out that's not an iWorks system. It's completely different than what I'm used to. The iWorks system, so instead of being a computer, it's self-contained, and it's not patently obvious which buttons to press to do what. So if anyone knows anyone who knows how to use that system, send them to me so I can get on my hands and knees and beg them and then try to bribe them with cold hard cash to help us out for that. Because the old system is right there, but Mitch said don't use it. You get a lot of noise with those leads. Part of the electronics has just gotten so much better in just 10 years. So yeah, I really hope we could. I'm just glad right now we have the belt now is fixed, that the treadmill, which was always the bane of our existence, actually works now. So we can do a proper Bruce protocol or a modified Bruce protocol, whichever one we choose to do. Yeah, no, great. That one I've used before. I know how to use it. It's the same system. And there's a video I can brush up on how to do that. But the EKG system, it's a new system and it's just a black box to me. So if anybody knows someone who knows how to use it, let me know. And I will do what I can to persuade them to help us out. All right, so tomorrow, don't forget, labs in 103. And we'll use microscopes tomorrow and lots of slides. There's lots of good material. We bought new slides, we placed them. And yeah, come and be amazed. Okay. Let's have a word of prayer. Father, thank you so much for this cold day. Please protect us from the winter storm we're apparently supposed to get. We pray for safe travel for those of us who travel. And we pray, Lord, that as your word says, it is your will that through our good works, we would silence the ignorant talk of foolish people. Lord, we pray that our lives would be thus. Through the power of your Spirit, in Jesus' name, amen. Okay, tissues are made of cells, so we need to do a little review on the nature of cells. Okay, are you going to cooperate this time? If I... Yes? Ah, looky. It actually worked. So eukaryotes all have internal membrane-bound compartments. The nucleus is one of those compartments. And the nucleus will house the genome, which consists of DNA. And in eukaryotes, that DNA is packaged into chromosomes. Chromosomes are these large linear molecules of DNA that are packaged into assemblages consisting of both DNA and protein. This combination of DNA and protein takes the DNA, which is quite long inside our cells, and compacts it very highly, and it's compacted into a structure known as chromatin. And there's lots of ways to stain cells to see the DNA inside cells. There are fluorescence stains you could use. There's this stuff called propydium iodide that binds to DNA and fluoresces like a Christmas tree if you shine low wave UV light on it. There are visible stains. Giemsa stain can be used. There's another fluorescent stain is called Hirsch, the Hirsch stain, which was developed in Germany, obviously German name. There's lots of different stains you could use to see DNA in cells. That's easy to do. So other membrane-bound compartments inside cells include lots of the structures you see here. We'll go over these. I don't want to belabor them right now. What you see in eukaryotic cells is that these compartments have a dedicated function. So the function inside the cell gets divvied up. Some textbooks will call this division of labor. I always thought that sounded like a political term. So specialization is kind of the way I think of it. So each of those compartments essentially has a job. So if you think of a cell, a prokaryotic cell would be essentially a little efficiency apartment with just one room. And you have your bedroom, kitchen, and bathroom all essentially in the same room. Very cheap, but not very pleasant. Whereas your eukaryotic cell would be a house with lots of different rooms, and each of those rooms is dedicated to a different function. You have the room with a boiler in it for heating. You have your washer and dryer in another room. You have a kitchen, you have a bathroom, you have a mud room, as I think we call it here in Michigan, where you take your shoes off when all the rubbish on your shoes from stuff like this and what have you, especially after it melts, and so on and so forth. And then you have the control center, the brains of the operation. That would be the nucleus in the case of the eukaryotic cell, because the DNA stores genetic information. That genetic information is expressed in the form of RNAs. And many of those RNAs are used to code for proteins, and those proteins will execute many of the functions inside the cell. So your book in chapter 3 has this really nice table that essentially describes many of these different types of membrane-bound compartments inside the cell. It also has things like chromatin, those of your assemblage of DNA and protein. Within the nucleus, there's also a very darkly staining structure. In fact, if you don't stain the nucleus, you can still see it because it's so dense. It's called the nucleolus, or depending on where you put the accent, nucleolus. Yeah, so either one, tomato, potato, whatever. This is the area where ribosomal RNAs are being transcribed at very, very high levels. And many of those ribosomal RNAs are undergoing processing, and they might also be assembling with ribosomal proteins for protein biogenesis that will then be transported to the cytoplasm to participate in protein synthesis. So the nucleolus is a very specialized structure inside cells. Fast-growing cells may have more than one nucleoli. The nuclear envelope, there's two membranes to the nuclear envelope. The outer leaf, well, not the outer leaf, the outer nuclear membrane is contiguous with the endoplasmic reticulum. And the nuclear envelope, as we will see, is decorated with pore complexes that will regulate what gets into the nucleus and what goes out of the nucleus. These are cytoskeletal elements that are going to help support and provide structure inside the cell. They involve the transport of materials inside the cell. Talk more about those. These support cilia and flagella that help cells move and what have you. Each of these are structures that we will get a chance to talk about. So peruse that table. That's a very useful table. Yeah, gotcha. Yeah, yeah. Vacuoles are also found in fungi. Vacuoles will be in lieu usually of lysosomes. So they are the lysosome for cells. So instead of digesting things, they tend to just store things because the plant cell is essentially hedging its bets. They're essentially doing what Jimmy Garoppolo should have done, just take the sack instead of just throwing it up and letting it get intercepted. They just store it because the cell is going to die. And when it dies, essentially, all that material gets thrown away. In fact, vacuoles are the reason leaves change color during the fall, because the vacuole acidifies. So the pigment inside changes color gradually as it acidifies. That's why the leaves change color. And then when the plants drop their leaves, they're taking the garbage out.
So like no, like you change the dynamic like every year. You don't have the temperature,
Yeah, soil dependent too. Yeah. Your evergreens, your conifers, things like that, they drop their needles throughout the year, essentially. So they're constantly dropping needles. So they use the same strategy, but it's not seasonal. Whereas with your flowering plants, it's based on seasons and so on and so forth. And you have regulated abscission and things like that. All right, so vacuoles are more for plants, fungi, things like that. Our cells use lysosomes. Let's take a stop at the nucleus. Inside the nucleus is DNA, deoxyribonucleic acid. That molecule stores genetic information. Genes essentially are stretches of DNA, and everything that's inherited is inherited because of DNA that you received from your parents. So if you have their eye color and their hair color, their hairline, the shape of their hairline, body type, even rough personality traits, things like that, those are all things that are inherited. All your human cells will have nuclei with some exceptions, the big exceptions being, of course, platelets, because platelets bud from nucleated megakaryocytes. And of course, mature red blood cells, they have nuclei at first, and then they lose them as they mature. And every nucleated cell will contain a full complement of the human genome that consists of those 23 chromosome pairs, which you see here. So when we talk about these paired structures, that is a chromosome. That is a chromatid. Chromosome, chromatid. So we have 23 chromosomes. We have 46 chromatids. All right. So if I ask you on a quiz, how many chromosomes do human cells have? You say 23. If I ask you how many chromatids do human cells have, you say 46. Thank you. Glad we cleared that up. The genome is housed within the nucleus, which is delimited by the nuclear membrane. So here's the outer nuclear membrane, and it is contiguous with the endoplasmic reticulum. So the endoplasmic reticulum will always be associated with the nucleus in some way. And there is a nuclear pore that decorates that nuclear membrane. And if you look at these nuclear pores in some detail using a freeze fracture here with a scanning electron microscope, you can see there are lots of those over the nuclear membrane. These are the doors for the nucleus. In order to get into the nucleus, you have to negotiate your way through those nuclear pores. If you want to exit the nucleus, you have to negotiate your way through those pores. These are the way you get in and exit the nucleus, or through those nuclear pores. You can see there are quite a few of them. Now, your nuclear membrane is a phospholipid bilayer, just like the cell membrane. Is this composition exactly like the plasma membrane? No, but it's still basically a phospholipid bilayer. It has a certain amount of protein, but it still consists of phospholipids arranged into a bilayer with some other stuff thrown in, cholesterol and stuff like that as well. Things that need to enter the nucleus have to go through the pore. Otherwise, the nuclear membrane is impermeable to most of the things in the cell. These nuclear pore complexes allow the passage of water and other small nonpolar molecules into the nucleus, but other proteins, other things, you want to get in the nucleus, you have to go through the pore complexes. Proteins that enter the nucleus have something called a nuclear localization sequence. That special sequence essentially helps them pass through the pore with the aid of a nuclear transport machinery. Here's a really nice transmission electron microscope picture of these nuclear pores. You can see their structure is pretty complicated. And essentially, think of a bicycle wheel with spokes, and the plug, you see plugs in the electron microscope. Many people think the plugs are something that's in the process of transporting. Most people think the pore actually looks more like this. And passage through this, again, is highly regulated. And your book essentially has this nice little picture of a nuclear pore complex. That's a very simplified picture, but that's essentially what it looks like. The passage through the pore is not automatic. It has to be able to have sequences within the protein that's going to allow it to get into the nucleus. The nucleus is also the site of gene expression. Gene expression. In order to express genes, remember genes, they're stretches of that DNA. You have to be able to access the DNA, slightly unwind it, and then you have to make an RNA copy of the DNA. So DNA stores genetic information. In order to express genes, you're going to make an RNA copy. RNA is an genetic informational intermediate. So DNA stores genetic information. You're going to make RNA. Some of those RNAs will have functions on their own. Others will code for protein. In order to express genes, you have to address the fact that the DNA is wound into this tight structure called chromatin. Chromatin consists of proteins known as histones. These histones assemble into an 8-protein complex known as a histone octamer. The DNA then winds around this octamer, much like the thread in a sewing machine winds around the bobbin. Please tell me you know what a bobbin is. Okay, Bethany's sitting there going like this. Can you explain to Evan what a bobbin is?
Oh, I'm gonna get, it's just like a whole extra, when we have a sewing machine, you have to fold one up and like down and above and below. So the bobbin is like the smaller one that it pulls from, like a smaller thread.
Right. It's a cylinder that's flanged, and the thread runs around there as it goes down to the needle, essentially from the spool. Is that fair, Bethany? You're far more sartorially expertise than I am. You struggle to say it. If I gave you a sewing machine, you'd do it in your sleep. But anyway, so these are kind of like little bobbins, if you will, or spools, if you will, around which the DNA is wound. They're then essentially clumped together into this very tight structure. That's how you get so much DNA in such a really small space. When you look at these cells tomorrow, some of you at least, others Thursday, you're going to see those nuclei are pretty small. Yet the DNA in those nuclei, if you were to lie it all out end to end, it'd be 1 meter long. You package all that into a really small space. Boy, if you could pack like that for vacation, imagine what you could take with you. It'd be pretty cool, huh? Well, that's due to the fact that you take these histones, you make the octamer, you wrap it around, and then you very tightly wind them about. Now, this tight structure up there, that's called heterochromatin. Can you express genes when DNA is wound like that? Survey says, yet. No. You have to unwind it. How do you unwind it? You modify the histones. Because histones are very positively charged. So because they're so positively charged, DNA, which is very negatively charged, has a natural affinity for it. So this binding of DNA to the histones, it's like peanut butter and jelly. It's like a king and a queen. It's like Joe Burrow and DeMar Chase. They like each other. Okay, they have an affinity for each other, essentially. So you modify the histones, you molecularly modify them. You put acetyl groups on them, you put other types of groups on them, you link those to them. Now the histones, they don't bind the DNA quite so tightly. And now they start to unwind. When they unwind, now you can express that DNA. You express that DNA, you pull those acetyl groups off the histones, they wind up again. So modifying the chromatin essentially is important to express those genes. So you make an informational intermediate, RNA. The enzyme that makes RNA from DNA is called RNA polymerase. RNA polymerase. In eukaryotic cells, like ours, there's 3 RNA polymerases. They were named by biochemists. They have the world's most boring names. RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Easy to remember, but really boring. RNA polymerase 1 makes ribosomal RNAs. Makes ribosomal RNA. And ribosomal RNA, you will remember, is the structural component and the enzymatic component in ribosomes that are used for protein synthesis. RNA polymerase 2 makes your protein coding RNAs. That makes your messenger RNAs. And then RNA polymerase 3 that's going to make your transfer RNAs. Transfer RNAs. And other small RNAs. Let's just say small RNAs. So those RNA polymerases, they will locally unwind the DNA. They will make an RNA copy of the DNA. It's single-stranded, so they're only going to use one strand as the template for the synthesis. They will make it from front to back, from 5' to 3'. Nucleic acids are directional. That RNA will either have a function of its own, like the ribosomal RNA or the tRNA, or they will be sent to the cytoplasm for translation, where they'll be translated by ribosomes into proteins. And that segment of DNA from which you make an RNA, that is called a gene. A gene. And that's G-E-N-E, not J-E-A-N. G-E-N-E. Now, another piece about RNAs from eukaryotic cells. RNAs from bacteria get made, and they tend to not have a lot of processing done to them. RNAs from eukaryotic cells are heavily processed. So they're modified at the front. They undergo a process called capping. You put a cap on the 5' end. That makes them longer lived inside the cytoplasm and increases the efficiency with which they're translated. At the tail end, they undergo a process called tailing. You have about 250 uridines that are added to the end. So you have a string of uridines. This is called polyuridination. And then inside the RNA, there are sequences that are removed. The sequences that are removed are called introns. And this process is called RNA splicing. RNA splicing. You remove the introns, right? You remove the introns, you take the remaining stuff called exons, you ligate it together, and that is your final RNA. This is actually important because, as it turns out, certain neurodegenerative diseases actually are due to defects in RNA processing. You don't make the right RNAs, and so your motor neurons die, and you lose the ability to move in some of these genetic diseases. So have any of you heard of spinocerebellar atrophy? Spinocerebellar atrophy, there's a spinocerebellar atrophy 1, spinocerebellar atrophy 2, 3. Turns out the 1 is due to a defect in RNA processing. Your motor neurons don't make the right proteins, they go belly up. Because they go belly up, your muscles can't move. And so that causes the neurodegeneration. And there is now a gene therapy that essentially lies across these RNAs. It's a synthetic oligonucleotide. You inject it, microinject it into the motor neurons. That small RNA lies across these and it helps those RNAs get properly processed. And this is past the FDA. It's now commercially available. It is ridiculously expensive, but because it's past FDA muster, the insurance companies will pay for it. And it gives hope to some of those children with these neurodegenerative diseases. Pretty cool, huh? Now, this complicates the definition of this term gene, because can you, theoretically, have a stretch of DNA from which you make an RNA, but you process it several different ways, and you make several different proteins from that same stretch? Yep. Yep. How many of you have heard of a gene product called the calcitonin gene-related protein? That is a target for migraine medicine because that protein goes way up in people who get migraines. That protein is made from the same stretch of DNA from which you make the hormone calcitonin. Two different proteins, two completely different functions made from the same stretch of DNA. You see the complication? So same one gene, two different proteins, very different functions. All right, we go outside the nucleus, we get into the cytoplasm. Let's first stop this. Anyone have any questions about genes, DNA, nuclei, chromatin, all that good stuff? Yes, Nana.
How does poly-U during the age of poly-A?
I actually got that wrong. Thank you for pointing that out. It's not a poly-U tail, it's a poly-A tail. I was thinking of the poly-Us that are used to isolate messenger RNAs because I was reading about those kits before I came to class. So I had that on the brain, so I got that wrong. That'll teach me to read those sites before I come to class. Polyadenylation, polyadenylation. Nana, thank you. Thank you. Yes, you add a string of poly-A's to the tail. Adenine, 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 blah, 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 blah. You add those at the end. A string of about 250. You have a protein called PAP, poly-A polymerase, that just sticks those right at the end of the messenger RNAs, that protects them and aids with their translation. That is put on the end of messenger RNAs, not put on the end of ribosomal RNAs or tRNAs or something like that, just on the end of messenger RNAs. All right. Thank you. Did you have any other questions? Okay, good. Thank you. Oh, you guys need to keep me honest. Okay, cytoplasm. Within the cytoplasm, you have these membrane-bound structures and some structures that are not membrane-bound. They're called collectively organelles. Organelle simply means little organ, little organs. So the membrane-bound ones include things like mitochondria, lysosomes, the endoplasmic reticulum, peroxisomes, and the Golgi apparatus. Others that are equally important, they're not membrane-bound, include cytoskeletal elements, microfilaments, microtubules, and immunofilaments, also ribosomes and centrioles and things like that. Let's talk about the plasma membrane first. That delimits the boundary of the cell. This is composed of a phospholipid bilayer. That doesn't mean that this is the only type of lipid in the membrane. There are other types of lipids in the membrane. This is a lipid that is definitely the most numerous. These types of molecules are the most numerous within the membrane, phospholipids. So when we talk about a lipid, we're talking about a molecule with a glycerol backbone we take these three hydroxyl groups and we link to them these fatty acids using the carboxylic acid part of the fatty acid. Now, these fatty acids, it shows the way that shows them here, they're fully saturated. These can be unsaturated too. And in fact, you find proportions of saturated, unsaturated fat. Do you know the difference between saturated and unsaturated fatty acids? Who doesn't? Huh? Right, there's a bend because there's a double bond. Right, and a polyunsaturated fatty acid has several double bonds in it. And that changes the geometry. The saturated ones are straight. The more double bonds you put in, the more kinks you put in. And if you have several double bonds now, they start to get, really, they look like a dead whoopee, essentially. Your whoopee dies in it. Never heard of that? Oh, anyway, so fatty acids, you get this linkage between them, this type of linkage. Okay, you're organic chemists out there. What kind of linkage is this? What kind of linkage is this? It says it up there. Ester, great gal, Ester. Okay, ester bond, ester bond. Now, if you have a glycerol with 3 fatty acids esterified to it, this molecule here is a fat, otherwise known as a triglyceride. So when you go to get a physical and you get a lipid panel, and one of those things that's reported is your serum triglyceride, that's what they're detecting in your blood, or how much of the fat you have in your blood. And that's usually, there are genetic conditions that can increase it, but it's usually the result of diet. So your serum triglyceride is a pretty good indication of how good or how poor your diet is. In most cases, again, there are people with genetic conditions that just have chronically high triglycerides. Your phospholipids are going to essentially replace one of these end fatty acids with a phosphate. That's why they are a phospholipid, as you see here. And then attached to that phospholipid is a so-called head group. That head group is usually charged. The phosphate's charged, the head group's charged, but the fatty acids up there, they don't like water. So you have a two-tone molecule. And it's kind of drawn this way, looking a bit like a transistor. This part hates the water. This part loves the water. You put a lot of these together and they're going to form a bilayer. And that bilayer is going to have the water-loving bits facing outside and inside, because the external part of the cell is aqueous. The internal part of the cell is aqueous, but the water-hating bits are going to face each other. And that provides the structure called a phospholipid bilayer. So here is the phosphate with the head group here, phosphate with the head group here, and the fatty acid bits, they face each other. And this is called a lipid bilayer, a lipid, or excuse me, phospholipid bilayer, phospholipid bilayer. Other molecules embedded in this, you have cholesterol. Cholesterol will regulate the rigidity of the membrane. Proteins, as you can see, are going to float in this sea of phospholipids, kind of sort of like icebergs. Proteins will play a really important role in that membrane in the life of the cell. They're important for transport. They're important for recognition. They're important as receptors for binding hormones and growth factors and other types of signaling molecules and things like that. They're important for adhesion and all kinds of other good stuff. So other types of lipids that you will have in the membrane. There's a group of lipids called sphingolipids. Sphingolipids will tend to cluster together with cholesterol in membranes, and they are going to usually cluster on areas where you're getting cell signaling. You can usually see these so-called lipid rafts. Lipid rafts. Because the sphingolipids, they're taller than the phospholipids. And so you can look at membranes using various type of technologies. You can see parts that all of a sudden kind of stick up. And those are these lipid raft areas where cell signaling is occurring. Water and small nonpolar molecules, they can pass through membranes. If you're small and charged or small and polar, forget it. You can't get through the membrane. That's one type of, yeah, we'll talk about transport. Facilitated diffusion facilitated diffusion uses a transporter. This would be passive diffusion. So things like, small things like ion, sodium, calcium, chloride, potassium, take a pick. Can't pass through membranes, charged. They bounce right off the membrane. Other small polar molecules like glucose, forget it. Bounce right off the membrane. So if you want to get through the membrane, you have to be either water or some sort of small nonpolar molecule. Otherwise, the cell is going to have strategies to take you up. Now, what if the cell wants to take up something kind of, say you want to eat something bigger than your head, so to speak. How do you do that? Oh, one bite at a time? Yeah, okay. Very good. The cell has a technique called bulk transport. Bulk transport. Bulk transport is used to take up things like LDL. Iron is taken up by bulk transport, so is vitamin B12. That's taken up by forms of bulk transport. One form of bulk transport is phagocytosis, where the cell essentially rearranges its membrane so it surrounds the object, the membrane fuses, and you make a so-called food vacuole, or a phagosome, as it's usually called, phagosome. Food vacuole is a very old term. It's usually not used anymore. A phagosome, and then that material in the phagosome is gradually degraded. There are only certain cells in your body that are capable of phagocytosis. Certain white blood cells, certain stationary white blood cells, reticular cells in the spleen, Kupffer cells in the liver, other types of stationary macrophages in bone marrow, they can do that as well, what have you. Because there's a certain machinery that's required for it. You have to be able to rearrange your cytoskeleton to be able to ruffle the membrane and get the membrane essentially around it like that. That material in the phagosome gets degraded and then utilized. Here's some pictures again from your book. You can see the extension of the membrane is called a pseudopod. The pseudopod is zeroing in on the ciliate here. And again, this is using cytoskeletal elements. This is a highly, highly regulated process. It surrounds it, dun, 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 essentially, and then it's going to fuse the two parts of the membrane with each other to make essentially a phagosome. That material in the phagosome will be gradually degraded, and anything that can't be degraded will be just kind of coughed up by the cell, much like a cat coughs up a fur ball. Either or. Depends on the type of cell, depends how fast it moves. Cells, neutrophils, for example, very active, very fast. They'll just kind of zip right around you. Macrophage is a little more deliberate about it, a little more calculating, a little more. So neutrophils are kind of more Patrick Mahomes about it, whereas the macrophages are more Tom Brady about it, more calculating, more surgical, more, you know. And Brady's retiring. I'm glad he's going to spend time with his family. He should. That's a more important thing. But I will miss watching him play. He was masterful to watch play, just masterful. So hey, I can watch him on old reruns. Now, endocytosis is another form of bulk transport. This is another way cells eat things that are bigger than their heads, so to speak. Receptor-mediated endocytosis requires a receptor that binds something very specific. And you get this pit, essentially, with these small particles bound to it. This utilizes a protein on the other side of the membrane called clathrin. I am running out of space here. So that structure there is called a clathrin-coated pit. The clathrin-coated pit, it's deforming the membrane. So the membrane gets deformed even more and even more and even more. It becomes like a flask-like structure. Then there are there's a machinery in the cell that'll fuse these two bits like that. You get this vesicle inside the cell. That's called an endosome. You can see it here, and then this is your endosome like that. You can see the vesicle is pinched off. The protein that helps fuse those bits of the membrane is called dynamin. Dynamin, it's like dynamite. There's a fruit fly mutation in dynamin. It's called Shibiri. There's a temperature sensitive allele of Shibiri. Because this process is so important for neurons, you can take the flies with the Shibiri, temperature sensitive Shibiri mutation, you put them on your hand. The second they get on your hand, they get paralyzed. They can't move. You can walk around the room with them. Second, you put them back in a vial and they cool back down. They get up and walk around like nothing happened. Most amazing mutation I've ever worked with. But that shows you the importance of dynamin. And then this can fuse with the lysosome and the components digested and what have you. So here's the vesicle. The clathrin on the outside dissociates from it, and it goes and does things. Cells can also non-specifically take up fluids and other large molecules. This non-specific process is called cell drinking or pinocytosis. Pinocytosis, and this essentially shows you some ways you do pinocytosis. You get a ruffle of the membrane that just kind of swings across, and you get something called a macropinosome, or you get other stuff like this. Again, this is a very non-specific, tends to be non-regulated process whereby cells can take up things that they just need. And I will let you take a look at this on your own. YouTube animation. Exocytosis, this is the means by which cells will get rid of things. This is also a process that is highly regulated. In cells, there is a process by which you regulate the secretion of particular molecules. There's another process by which the secretion just occurs all the time. It's called constitutive secretion. And notice that the Golgi apparatus is going to play a role in this as well. This process whereby these vesicles fuse with the plasma membrane, and then you dump their components into the extracellular environment. This is very much how neurons release neurotransmitters. Exocytosis is mediated by a machinery called the exocyst, not exorcist, exocyst. And this again can be very highly regulated, or it could be something that the cell's just always doing. In the case of neurons and the release of neurotransmitters, of course, that process is very highly regulated. Cilia are extensions of the plasma membrane. We'll see some of these in our slides tomorrow or Thursday. These have microtubules that are anchored in what's called the basal body. They extend and the membrane surrounds them, so it's an extension of the membrane as well. There's many cilia in cells. They also beat in a coordinated fashion. So they beat kind of like oars on a biking ship, stroke, stroke, stroke. Essentially, they all go in the same direction, or they go in a coordinated fashion where the cell does the wave, like that. So it's very coordinated, highly regulated. It's not just haphazard, oh, let's just go at it. No, it's very highly regulated, highly coordinated. Here's some cilia. This is a section and scanning electron microscope of a fallopian tube. And fallopian tubes will have cells that are ciliated, cells that are not. Some of the non-ciliated cells or secretory cells are called PEG cells, PEG, PEG cells. They make mucus and other things. And the cilia, you can see they look like little hairs. And that's kind of sort of what they are. Now, flagella, at least in our cells will have one flagellum usually. And whereas cilia are short, flagella are long. And they move back and forth in a whip-like fashion, like this. These are also microtubule mediated, and they're essentially used to propel things, usually spermatozoa. And they propel that spermatozoa very quickly. The structure inside the flagellum, very similar to cilia. So mutations that affect cilia will also affect flagella. Other extensions of cell membranes are examples like microvilli. Microvilli don't move. They essentially increase the surface area for absorptive purposes. These are also supported by cytoskeleton, but these are supported by microfilaments rather than microtubules. Microfilaments are made of actin, microtubules are made of tubulin. So we see these in the digestive system, we see these in the kidney, we see these in places where you have absorptive surfaces. And that brings us to the cytoskeleton. When you think of cytoskeleton, I want you to think of concrete and rebar. Rebar. Who here has done some construction? Anybody? Evan, what's rebar for? Exactly, exactly. There was, where was it? Minnesota or Boston or one of the places somebody tried to skimp on the construction. They were redoing overpass. They didn't put, no. They put rebar in, but it was really low quality steel. And the forces essentially just shanked it right in half, and the whole side of it essentially came down. So the rebar was compromised, and the whole concrete just went essentially like your mashed potatoes. If you know, when you're, okay, I did this as long as a kid. I tried to make a wall of my mashed potatoes and I always went because my mother never made them very stiff. So I tried to build up the wall and the wall would root. That's what concrete does if it doesn't have rebar. I should have taken the string beans and put it in the concrete, put it in the mashed potatoes. Bethany, I was a weird kid, okay? Just, your kids might be weird too. You never know, you might marry a nerd, who knows? Just get over it. Yeah, I've known Bethany for a long time. She's friends with my daughters, and so I can play with it like that. I was her quiz coach, and she already knows I'm weird, so yeah. Rebar is going to reinforce that concrete, so it doesn't essentially do the mashed potato thing. These cytoskeletal elements do very much the same thing inside cells. They provide structure inside cells. Vesicles are transported along cytoskeletal elements. They also provide the cell the means to resist shear forces. This ability to resist shear forces is very important in the life of the cell because cells often are going to be subjected to a fair number of shear forces. So there's three types of cytoskeletal elements. There's microfilaments, which are polymers of actin. There are microtubules, which are polymers of tubulin. And then there are intermediate filaments, which are polymers of intermediate filament subunits. There's different types of subunits. We'll get to those. Microfilaments, microtubules, and then the intermediate filaments. Here is a very beautiful staining of cells using an antibody against tubulin. See the hole right here? What do you think that hole is? Oh, someone said it. Someone said it. Nucleus. Someone said nucleus. Who said nucleus? I heard someone whisper it under their breath. That's the nucleus. There's no tubulin in the nucleus. If you wanted to double stain this with propydium iodide, which is excited at about the same frequency as the laser you'd use for this, this would glow blue, this would be green, and it would look really stunning. Now, if you look at the tubulin, that's all over the bloody cell, isn't it? This stuff is really widely distributed throughout the cell. This isn't just found in one little place. This is all over the place. This provides a really important function inside the cell when it comes to maintaining the shape of the cell, helping the cell move, helping the cell change shape, attach, and all that stuff like that there. Essentially, vesicles that are moving along these, the internal structure of the cell, that's all the function of the cytoskeleton. Really, really, really important in the life of the cell. Organelles within the cell. Organelles will often attach to various cytoskeletal elements and they'll move around the cell. In 112, I think, those of you who took 112, you did an experiment where you looked at a plant cell and you saw the plant cell do this. That's called cell streaming or cyclosis. That's a cytoskeletal induced phenomenon. If you take the cytoskeleton, you disassemble it using various drugs, that'll stop. Cell movements possible because of the cytoskeleton. Now, intermediate filaments are very interesting because if you grow cells in culture, they'll stop making intermediate filaments. Once, however, you start subjecting those cells to tensile forces, they'll remake them. Now, in your skin, you have an intermediate filament called keratin. Keratin is a type of intermediate filament. There's a genetic disease that involves mutations in keratin. It's called epidermolysis bullosa. You could do this to someone with epidermolysis bullosa. If you did, within the next few hours, large swaths of their skin would start to fall off because their skin cannot resist shear forces. And there's been a tremendous amount of genetic work done with this type of disease, and it's clear that keratin, the role of keratin, is for skin cells, keratinocytes, to be able to resist shear forces. That's what these intermediate filaments do. They provide the ability of cells to withstand shear forces. Cells in culture, they don't really have shear forces. They don't need them. They stop making intermediate filaments. The second you force them to withstand shear forces, and you can do that by growing them on cultures that will do this, so they get stretched. They'll start making intermediate filaments because they can't survive without them if they're subjected to shear force. Yeah.
What does the magnitude of the force have to be for it to like affect the skin?
With people with that mutation,
Yeah.
depends on the allele that they have. There's 7 alleles, some are more severe than others. The magnitude for people with the less severe types, you want force like in dynes or something like that, or just
Well,
kind
I'm
of,
just curious because, like you said, like just rubbing a skin to like any sport.
oh, well, clothes. Clothes, actually, certain types of clothes can actually cause their skin to start to fall off. And so they have to wear very baggy clothes. They can't wear anything too tight, or it'll actually cause their skin to kind of slough off. Yeah, it's, there's gene therapy in the works for individuals with that condition. The problem is you have to engineer large swaths of cells. There's also a procedure where you can take cells. It's possible now to grow cells, skin cells in culture. When you grow skin cells in culture, you can genetically engineer them. The goal is to take this genetically engineered skin and then grow it in large swaths, peel the skin off, and then replace it with the genetically engineered skin. That's what's done for burn patients now. You take sections of skin, you grow it in culture into large swaths, and you replace. Now, provided you can get the burn patient to survive that long, because they have to deal with dehydration and massive infections and stuff like that. Burn units are very good. In fact, Gotcha's going to be a nurse. Burn unit nursing is a very specialized area of nursing that takes special training. And burn units are if you have a burn unit nearby, they're very good at getting burn patients to survive. You know what the other thing that's used for burn patients, because you have to remove the dead skin. You know what's really good at removing the dead skin? Maggots. maggots. Several burn units now have sterile cultures of maggots. You put the maggots on, there's no inflammation, there's no infection. The maggots, in fact, make little antibiotics in their saliva, and then you could put the skin right on and just as slick as Willie.
Does the mutation only affect like certain areas or the whole entire body?
Some alleles affect some areas more than others. So there's the Lewis allele, for example, will tend to affect intertriginous areas, places where the skin bends because of joints. The more severe alleles will affect there's one that just affects the face. So shaving, you can't really shave if you have that allele, otherwise, oh, darn, there goes my skin. It depends on the allele. And genetically, it's actually very interesting. There was a review published on that in annual reviews of human genome genomics and population genetics. If you're interested in it, I could give the resource. But that's very, very interesting piece of research now that's being done in several areas. Helen Blau, B-L-A-U, she's kind of the mover and shaker behind a lot of this research at Stanford. She's been one of the pioneers in the genetics of epidermolysis bullosa. Yes, but they have to wear special gloves that won't stress their skin. Yeah, they do. Right. So if we look essentially at, I really like this picture from your book because it essentially shows how the organelles in the cell are supported by this network of cytoskeleton. And they essentially don't really go anywhere unless the cytoskeleton allows them to. And this, I think, really underscores the importance of cytoskeleton. Lysosomes. The lysosomes are the garbage disposal of the cell. They have a cadre of enzymes called acid hydrolases. So these lysosomes, they fuse with endosomes. They will then acidify the content of that lysosome endosome structure. The acid hydrolases then become activated and they degrade proteins, lipids, complex carbohydrates. They degrade them to the more basic elements. The cell then uses them as energy and food sources. This strategy essentially guarantees that these enzymes are only active in a lysosome that is actually working. If the lysosome bursts and the cells find themselves in the neutral pH of the cytosol, they won't be active and they won't degrade the cell. Here's a nice picture from a transmission electron microscope of lysosomes. You'll notice lysosomes tend to look really electron dense. That's really common for lysosomes. Also, lysosomes tend to be in close proximity to another vesicle called a peroxisome. Peroxisomes are used by mammalian cells, at least, to degrade unusual lipids, also to deal with certain cell toxins like hydrogen peroxide and superoxide radicals and things like that. So they are detoxifying vesicles. That's why you usually find them in association with lysosomes, because lysosomes tend to generate a lot of rubbish. Also, things that lysosomes can't handle, they just send them to the peroxisome and the peroxisome will do them in, so to speak. Okay, that is enough for today. We'll go to mitochondria, finish this up, go on to the next section, look at the mitosis. I'll see some of you tomorrow for lab, 8 o'clock. Please be on time. Have you all picked up your lab manuals? They are in the office. Mr. Schaefer isn't in today. I'll tell you what, I'll toddle on down there and I'll take your money and so you can get your lab manual. Or I'll just give it to you and you can pay for it later.